Good. So let's, you can start. Thank you.
Okay. Well, thank you to you. So I will be talking about recent work that was done in collaboration with Yakov Kiorin and Rama Ranganathan on the coevolution of proteins. And so the general goal is essentially to understand proteins, how they work, how they evolve, and more specifically to understand the relation between the sequence of a protein and its function. And by function, I mean broadly all its biophysical properties as well as evolutionary properties. And so one of the difficulties is, as you probably know, that amino acids do not contribute independently to the functions. There are complicated interactions. So this is also known as epistasis, which appears as the fact that mutations have an effect that depends on the background of what the other amino acids are. And in the context of proteins, this is particularly difficult to understand because the base data we have is structural data, that is the knowledge of the relative position of amino acids in three-dimensional structures. And the patterns of epistasis are not obviously related to the distance in the three-dimensional structure. And in particular, we have what I will call long-range epistasis, which correspond to the fact that you may have positions that are distant in the structure, but are nevertheless coupled epistatically. And so this is what is behind the phenomenon of allostery, long-range regulation. But it's also well known in the context of enzymes that you have mutations far from the active sites that have nevertheless a significant effect. And so one approach to this problem is to look at evolutionary data and more precisely to study multiple sequence alignments of protein families. And there have been many methods analyzing this data. I'm going to mention only two of them. The first one, which is known as SCA, or statistical coupling analysis, was designed precisely to try to identify this long-range epistasis. And so it was developed by Rama Ranganathan and collaborators over the years. And the main finding is this concept of sector, which is represented on one example here, which is the idea that you may explain this long-range epistasis by the presence of a network of coupled amino acids that go through the structure. And so for instance, in this example, this is represented in blue. And this has led here to the prediction that the site that is marked with a red star is an allosteric site, which was later on verified experimentally. But here I'm going to discuss this question of long-range epistasis in the context of a different framework, which is known under different names as statistical coupling analysis, maximum entropy models, or POTS models, which is a framework that was developed by Martin White and collaborators a bit more recently. And so this was, I think, already introduced by Rémi Monasson on Monday, but I will just remind you the basics before coming back to this problem of long-range epistasis. So the starting point of this method, as the other method, is a multiple sequence alignment of a protein family from which we collect statistics at the level of individual positions, so the statistics of amino acid at individual positions, and the statistics of amino acid at pairs of positions. And from this information, we built a statistical model. So the P of S, which, if you wish, represent the probability for a sequence to belong to the family. And it has a Boltzmann form where the energy is the fitness, that denotes phi. And this phi is taken to be composed of two kinds of terms, terms that are couplings between pairs of sites, this is Gij, and terms that are called fields that are at the level of individual sites. And these parameters are defined essentially to reproduce the statistics that we have in the alignment, this I phi A and Fi J A B. And so one reason to be interested in this model is that it has been shown that they encode a lot of relevant information, starting from the observation that if you look at the pairs with largest couplings, they typically correspond to pairs of sites that are in contact in the three-dimensional structure, even though they are not necessarily proximal in the primary sequence. So this is the first indication that it contains structural information. It also contains functional information, and this is what Remy was presenting on Monday, the fact that by looking at how this function phi change when we make a mutation in the sequence, we can correlate this change with the experimental examination of the effects of mutations. And this has also been demonstrated in a number of cases. And finally, more recently, the model was shown to be generative in the sense that new sequences that were drawn from this distribution P of S were shown experimentally to be functional in vivo. So it seems that this model contains all the information that we may want, but there is still a puzzle that I would like to address here, which is the fact that there is no obvious indication of long-range epistasis. Okay, so there is no large coupling Gij between two positions that are distant in the three-dimensional structure. And this, in spite of the fact that there is extensive evidence for such effects, and also in spite of the fact that the other method that I was mentioning is able to find this information in the alignment in the form of correlations. Okay, so is it really a puzzle? Because one may say that physical interactions are mostly short range, and this long range epistatic effect may come just from chain effect through the indirect contribution of short range interactions. And all these post models are in fact designed to disentangle the couplings from the correlations. This is why they've been called direct coupling analysis. But I mean, each of these statements is correct, but we can still not conclude that we should not see long-range epistasis in the form of large couplings between distant sites. And the reason is that, I mean, physics may be short range, but here we are talking about a function that is defined in sequence space. And if we accept that this phi is a fitness that can score mutational effects, having no couplings means that we are predicting that there is no epistasis between these two sites. And more generally predicting that these couplings are weak, weak compared to the one that correspond to contact, then the implication is that the epistatic contribution is also much weaker. So what could be the solution to this apparent paradox? So one may be that, okay, this long-range coupling exists, but they may be a minority, and we have just been focusing mostly on the top couplings corresponding to structural contacts, and maybe it's just a matter of looking more carefully to these top couplings. And so this is something that has been done systematically by Anishenko and collaborator. And so what they found by analyzing several protein families is that essentially in every case where there was a top couplings for which there was not an obvious interpretation in terms of contacts, in fact, there was still an explanation based on short-range interaction, either because the protein was actually a normal oligomer where the coupling corresponded to the interface between different domains, or maybe there was in the alignment an alternative structure that was consistent, or it was in some cases just an error in the alignment itself. So the conclusion that there is no evidence for strong coupling between distant sites. And in fact, they even looked at cases where we predict this sector, this network of co-evolving amino acids that are supposed to explain this long-range effect. And what they show is that there is not even an enrichment of large coupling inside these networks. So what the conclusion that they give is essentially that, I mean, if there is any long-range coevolution, it has to be much weaker than the short-range coevolution. And obviously, this is assuming that, I mean, the model is correct. So another possibility will be that there is a problem with the model, or maybe there is a problem with the interpretation that we are giving, particularly of this function phi. But so what I would like to suggest today is that there is actually a fourth possibility, which is that all of this may be explained by a statistical artifact. And so to explain it, I'm going first to demonstrate this statistical effect in the context of a very simplified toy model. And then I'm going to show you that the data actually looks like the toy data that we obtain from the toy model. So what's the principle of this toy model? It's very simple. It's just to define ourselves a model that by definition is a post model, where I will set myself the couplings GAJ and the fields HI. I'm going to sample sequences from this distribution. So this will be analog to the multiple sequence alignment. And then I'm going to apply the usual method to try to infer back the parameters of the model. And so what I want to show you is that in doing so, in some way, I'm going to recover the short-range couplings, but somewhat the long-range couplings are going to disappear. And so here is the definition of the toy model that is on purpose extremely simple. So it's a model where, okay, I have a sequence of length 20, I have like 10 amino acids, they are all equivalent. There is no field, it's purely couplings, and the couplings are either 0, represented in blue, or they are equal to 2, represented in red. And so what we are trying to capture with this model is the fact that there are different patterns of couplings. You can either be coupled just in a pair, okay, like in the upper left part, and this is what we will interpret as coupling between contacts, or you can be involved in a network of coupled amino acid, which is what we will represent here as a sector. And here I'm showing 2 of them with different size, so that you will see a bit of the size of these networks matters. So from there, the idea is just to sample, and this can be done simply by Monte Carlo sampling. And I'm going to sample, in this case, 300 sequences. And this is a number that is chosen so that it's small in comparison to the number of parameters that I have in my model. And this is comparable to what we have with real data, where we also have relatively few sequences in comparison to the number of parameters. And from there, I'm going to apply a standard inference method, which is maximum likelihood, that I will use with regularization, L2 regularization, which is precisely to address this problem that I have very few samples in comparison to the number of parameters. And so this is, if you want, the formulas for this log likelihood, where I have this regularization parameters lambda, that are controlling the penalty that I put on the large couplings or large fields. Okay, so what do we get? So here is what we get with relatively small regularization. And what you see is that we get a very biased view of the original model. So if you look at the top couplings, they are in red. They do represent this isolated coupling that I had on the top. But if you look at the one that belong to the large collective unit, they are very weak, and they are in fact weaker than couplings that should be 0 but appear to be non-zero, and that you show here you see here in yellow. Okay, so this is the idea that you may have this coupling, but somewhat they disappear through the inference. So what happens? So the problem is not the method itself, but it's really the fact that we are undersampling a lot. So this can be shown by just varying the number of sequences that I'm drawing and that constitutes the multiple sequence alignment. And so what I'm showing you here is what happens when I increase this number m of sequences. So, and each curve corresponds to an average of the coupling for the different types of patterns. Okay, so in black, this is averaging the contribution of this isolated pairwise coupling. In red, you have the contribution coming from the largest unit at the bottom, and in blue, the one in the middle. And so what you see that there is something that looks like a resonance at a particular value of the size of the alignment. And in particular, if you look at small sizes, so this is dotted line that you can see that correspond to what I was showing before, you see that you are emphasizing the contribution of the isolated pairs and somewhat missing the contribution to the larger patterns. But if you choose a different size of the alignment, you may see something different until you get to very, very large value where you get the exact results where everything converges to one. So, but the problem obviously is with real data, we just have a finite number of sequences, so we cannot play with this parameter. But there is another parameter with which we can play, which is this regularization parameter. So this is lambda j. And what we can see is that by varying this parameter, actually, we can reveal different types of patterns. So on the left, you have what I've shown you in the first graph, small regularization. But as you see, as you increase first, you will get mostly the small collective units until you get to very large value where you get where you have a situation where you are dominated by the largest coevoluting unit. So what do we learn from this? I mean, so the basic effect is that when you have patterns of couplings at different scales, and when you are in a regime of severe undersampling, then you typically get a biased inference. You can somewhat control the bias by playing with the regularization. This is this figure. But you can also see that there will be no regularization at which there is no bias at all. And finally, you can also understand that what you see at very large regularization is essentially proportional to a correlation matrix, which is also consistent with the fact that other methods based on correlations actually detect primarily the large coevoluting units. So this is a toy model. So what can we say about real data? So in this case, obviously, there is no ground truth for Gij, but what we can do is we can play with this regularization, as I was showing. And so this is what I will demonstrate on this example of Curie's mutase, which is the protein on which the synthetic design was recently done. So this is a case where we have every reason to think that these POTS models are good. And so briefly, these proteins are enzymes that when you look at the structure, they come in dimers. Okay, here you have one in gray and one in blue. And so when you make this inference with two different values of the regularization, one very small, one very large, you find again that different positions contribute in one case or the other case to the inferred coupling. And so here I'm showing the result where I have ordered the position in such a way as to highlight these differences. So I've ordered the position as to the extent to which they show more significant couplings with low regularization or high regularization. So there is no true couplings again which we can compare these results, but we have the structures, and so we can ask where are located the different positions that we find here. And so what I'm showing here is represented on the structure, the top 20 pairs that contribute either to the result at low regularization or high regularization. And you see that for high regularization, you get essentially positions that are in the core of the protein. And this is very much consistent with what you find, what you can define as a sector, and also what experimentally are the positions that are the most important for the function. And the one that you find on the opposite limit of small regularization are more on the periphery, represented in blue here. So these results are consistent with the fact that there are these two different scales in the problem that we can recover in this way. And what's interesting is that so the limit in which this method is usually used is this limit of low regularization, okay, where we find these contacts. When you apply it here with high regularization, so the yellow positions, you get again contacts. You actually get new contacts, but in addition, you also get pairs that are coupled but not directly interacting, and are candidate for representing this long-range couplings. So, okay, so that's essentially what I wanted to present today. So the idea that when we are trying to interpret the apparent absence of large coupling between distant pairs in three-dimensional structures, we should be aware of this statistical effect, which is again the fact that when you have these different scales and a very limited sampling, you typically get a biased inference. And so this could explain why we don't see those couplings even though they are present. And just to finish, I wanted to mention that, so if you accept the presence of these different scales, there is also the problem of understanding more generally how they are related to the different functional properties that I introduced in the beginning. And here, this is also a good problem that we can address using toy models. And this time, toy models in which there is physics and evolution. And so I will not say more, but if you are interested, this is partly published, while the first part actually is still unpublished. And so it's mainly the work of Yakov Kliorin. Yeah, that's, so I will stop here and thank you and take your questions.
Or hide them directly. So I see a first question from that phylogeny models are discrete. Is there references for the continuous one defining the coevolution? You can read it in the chat. I'm not completely sure I read it well.
So here actually, I mean, we are not reconstructing the phylogeny. You may have noticed. So yeah, I don't know if this is the question.
Okay.
So and I mean the model are discrete because yeah, amino acids are discrete. This is for sure.
There is a question by Martin Weid. Is there evidence that you find long-range couplings in very large MSA, e.g. 10 to the 6 sequences? Would models like RBM with collective modes would help?
Okay, so the first question, so we haven't looked and we haven't found, but you may have to go far beyond 1 million sequences, I would guess. And then, so would different models help? I mean, they may help in the sense that even here, you know, using different regularization, you can find this large units. But I mean, the basic problem is because you are undersampled, you have lost essentially the information about the relative scale of these different couplings. And so you need somewhat to introduce some hypothesis, and it's going to be very difficult to recover correctly all these relative scales if you don't treat differently these different patterns. So here, if, for instance, if you don't have a different regularization that is associated with the different pattern.
May I explain why I asked this question? Because in restricted Boltzmann machine, like Remy had in his talk, you have explicitly these collective modes, And these collective modes, so you have for I don't know, your little square in the upper in the lower right corner, you would have just something which has very few parameters as compared to having many couplings. So in principle, you would expect that you need much less regularization.
Okay, no, this is possible.
So,
yeah,
I don't know.
We haven't looked at yeah. So I think this is yeah a matter for future work to see to what extent different methods can address this basic problem.
Okay, Henri.
Yeah.
Thanks.
Can I ask one question also?
Yeah.
So a long time ago, we also developed another alternative model based on Bayesian networks, whose one advantage is that there are no overfitting or no regularization because everything can get. So I would be very curious whether these artifacts also appear in those models, because in your case, you can still say, well, you know, you have too many parameters and then you have to regularize and it's causing it. So I would be very curious to see that. I'm actually wondering, why do you think that evidence for long-range epistasis implies evidence for long-range Jij that are not zero?
Well, so it's just, so again, here. Okay, so this is the idea that, I mean, if you take this function phi to represent the fitness by which you score the effect of mutations, okay, so, and you compute a delta phi, okay, which will be the effect of, I mean, comparing the effect of 2 mutations to the additive effect of single mutations, then you will get this coupling. So it's directly, I mean, there's a direct relation between this coupling and the delta phi based on position ij.
Okay.
Okay, there's 2 last questions. Let's take 2 more questions. So there is a question by Anne-Florence. You want to ask it directly?
Sure, yeah. So this was a follow-up actually to Martin's question. So first of all, thanks a lot for the great talk. I would like to ask whether, you know, in fact, you need to go beyond 10 to the 6 sequences to get these long-range signatures, because in reality proteins Curie is 20, or is it a different reason?
Yes, so, okay, so this 1 million, okay, it appeared here, I mean, the context of this very simplified toy model.
Right, yes.
But yeah, so it will scale, so what you can see that it's scaled with Q square, and it also scale with log L. But it's also scale, I mean, it will also depend on the, you know, the strength of the couplings and everything. So this is what I was saying, that I'm expecting that with real alignment, you may need far more than 1 million sequences.
Okay, thanks a lot. So, yeah.
My 1 million was basically the largest alignments you find in UniProt and so on.
Right, yeah. So that would be super interesting, yeah. Thanks.
Okay, so let's take the very last question by Andrea. With large regularizations, can you reproduce 1 and 2 point correlation functions of the teacher model with the student model you obtain? Well,
so yeah, I mean, if you use very strong regularization, you will get a very distorted model, and you will not fit very well your correlations. But as usual, you have to be careful, because with low regularization, you will fit very well, but you will overfit. And so, yeah, so the good fit is not the only criterion to judge the quality here. And since we are talking about unsupervised problem, it's actually a non-trivial matter to find optimal regularization, even though I also say that here there is no evidence, when we know the real truth, that there is any optimal regularization.
Okay, so thanks a lot. And it's time to hear Clément. Clément Isaac, can you